Welcome back, everybody. So, in this chapter, we are going to focus on metabolism and nutrition. So, what is metabolism? So, it is all of the chemical reactions that can occur within an organism. And when you talk about cellular metabolism, we're saying the sum. It is a sum of all chemical reactions that can occur within our cells. And this is important because this is how the cells get the energy that they require to perform their essential functions. Now, there are different types of reactions that, of course, are involved in metabolism. There are two major ones that I want you to know: catabolic reactions versus anabolic reactions. So, anabolism involves what? The synthesis of organic compounds. You're making organic compounds from smaller fragments. Okay, so anabolic reactions involves building organic compounds. All right, a perfect example of this is ATP. ATP is a molecule that can be built, and it is usually created by what? The combination of inorganic phosphate with ADP plus adding energy into the system will build ATP. Now, the second type of this process is what we call the catabolism, or you can say the catabolic reaction. And what catabolic reactions here is you're breaking down organic compounds. Are you following? And a perfect example of this is also ATP. We can do this with ATP. We can do this in the reverse reaction where we're going in this direction. Okay, so this double-headed arrow tells me that this equation here, this chemical reaction is reversible. So I can build ATP in this direction, or I can break down ATP in this direction. So this is why we have the double-headed arrow here, okay? So the catabolic reaction is when ATP is broken down into ADP, all right, and inorganic phosphate. And the breakage of this phosphate bond releases energy into the system. Are you following? So catabolic reactions, organic compounds are broken down. Okay, and a lot of times that energy that is released is roughly about 40% of the energy is then used to now make ATP again and to carry out cellular functions. Okay, but 60% will escape into the environment and warm up the interior of the cell and surrounding tissues. Now, another type of reactions that we see in metabolism is the redox reactions. So the redox reactions usually involve the gain or the loss of electrons. So if we are gaining electrons, it's a reduction reaction. Remember that. Reduction, I know. It's kind of opposite when you think about it, but a reduction reaction in chemistry is a reaction that involves the gain of electrons. Oxidation reactions involve the loss of electrons. Are you following? So here's a perfect example. When we look at these molecules called NAD plus and FAD plus, what are they? They are coenzymes. They are coenzymes that are important for transporting hydrogen electrons in the cellular respiration process. So this is going to be important in cellular respiration where we generate ATP for the cell. So these coenzymes, they can undergo reduction reactions or oxidation reactions. So I'm going to give an example, okay? So here is NAD plus, okay, in its oxidized form. And suddenly here comes a hydrogen electron. Yes, this is how we denote the hydrogen electrons. Notice here, when the hydrogen electron combines with this coenzyme NAD+, it provides you with the reduced form of NAD+, which is NADH. So NADH has been formed by the reduction process. Are you following? Where you added an electron. So NADH is basically the form that gained a hydrogen electron. The same thing can happen to FAD plus. FAD plus usually carries two electrons. When you see it in its reduced form, it usually looks something like this. Okay, it carries two hydrogen electrons, and we'll see this later. But just know that this is the reduced form of it. Okay, when they have gained electrons. Now, what about the opposite? When they have lost their electrons that you're usually carrying? Well, notice this arrow here is a double-headed arrow, so it tells me that this reaction is reversible. So if we are going in this direction, like that, what's taking place here? The NADH in its reduced form, it is going to be oxidized. And when something gets oxidized, it will lose its electrons. So in this case, notice here, we have broken down NADH into what? Its oxidized form, NAD plus, and the hydrogen electrons. Okay? So be prepared to answer questions like this. Know this example. It doesn't matter which way I put the arrow. Know which direction is a reduction reaction versus the oxidation reaction. So this concept, the next concept here is actually a summary of how our body, how our cells will break down, these organic compounds and utilize them for energy. This is just a summary, and I want you to read through it and just be familiar with it, okay? So now we're going to begin with carbohydrate metabolism, the digestion of carbohydrates, and also the absorption of the monosaccharides after digestion has occurred. And how does the body utilize carbohydrates for energy? So we're going to go through the process, and you will notice that this information is similar to what we covered in chapter 23. So if you have studied this in chapter 23, you should be okay for chapter 24. This should be a review for you, okay? Only few information in here will be brand new. So let's begin. Now we remember from chapter 23 that carbohydrate digestion actually begins in the mouth. Remember that, okay? And how does this occur? Well, the chewing process occurs within the mouth, which is mastication. While this is happening, you have the bolus within the mouth, covered with salivary secretions. And part of the salivary secretions are your salivary amylase. Remember, this was the enzyme that helped to break down complex carbohydrates within the mouth. So it will start to break down all of your polysaccharides, all of these carbohydrates into disaccharides and oligosaccharides. Okay, so we're going down. We're going down the list now where we're taking it down into smaller components, but we're not there yet. But in the mouth, this process will begin, and it's usually done by salivary amylase. Make sure you remember that. Now, salivary amylase and this bolus will then, be transported through the esophagus down into the stomach, and we know something will occur in the stomach. Salivary amylase will lose its function within the stomach as soon when it arrives there, and here is the reason. Remember, the high acidic conditions within the stomach will cause this. Okay, the pH of the stomach, remember, it can drop very low, all the way to a pH of 2. Remember that. And when that happens, carbohydrate digestion stops. Are you following? So carbohydrate digestion begins in a short period of time as soon as it enters into the stomach, but it doesn't continue as long. Okay? That whole process is shut down because salivary amylase is denatured due to the high acidic conditions of the stomach. Are you following? Now, after the stomach, where do we go? We go into the small intestine, the first segment of the small intestine known as the duodenum. Now remember, remember we call what has come into the stomach chyme. After it is mixed up with all the gastric juices, we call it chyme. So when that chyme, that carbohydrate-rich chyme, enters into your duodenum, what happens? Well, this signals the duodenal cells to release the hormone called secretin. Are you following? Now, secretin, make sure you know these hormones. Listen, every enzyme listed, you need to know its function and its role, okay, in the digestive system. Yes, this information will be tested, so you need to know the function of these enzymes that are listed in bold, and you need to know the function of the hormones that, play a role in this process, okay? All right, so secretin, released by the duodenal cells, will then go and stimulate the pancreas. Are you following? To release sodium bicarbonate. Now, sodium bicarbonate will be important to buffer the the pH of the chyme within the duodenum. All right. This will ensure that all the enzymes that are going to be doing their work in the small intestine, they can perform well without being denatured. Are you following? So remember, acidic chyme comes in from the stomach into the duodenum. And that could be a problem because the duodenum is not built to handle that acidic chyme. So we need to neutralize that acidic chyme that have entered into the duodenum. And this is done through what? The secretion of sodium bicarbonate from the pancreas. And the pancreas will release this under the influence of the hormone secretin. Okay? At the same time, CCK, or the hormone called cholecystokinin. Cholecystokinin, make sure you know the name. Cholecystokinin, abbreviation of CCK. CCK, which is also released by the duodenal cells, they will go and stimulate the pancreas to release pancreatic amylase. Now, what is the job of pancreatic amylase? It is similar to the salivary amylase that was secreted in the mouth. Well, pancreatic amylase will continue the breakdown of what? Of your carbs. Okay, so it will continue the job that salivary amylase started off in the mouth. So the carbohydrates will be further broken down, into more disaccharides and oligosaccharides. Are you following? And a lot of times, because that chyme contains a lot of carbohydrates, the release of GIP, gastroinhibitory peptide, will then stimulate the release of insulin by the pancreas. And we know the job of insulin. Insulin will help to do what? It will help to move glucose out of the blood and put it into the cells where they need to be and where glucose can be used for energy. Okay, so take note of the hormones that have been released as a result of that acidic chyme entering into the duodenum. Okay, now as we go further down into the small intestine, we're now going to encounter the brush border cells. The brush border cells. So I hope you remember who the brush border cells are. These are your simple columnar epithelial cells lining the walls of the small intestine. But majority of these cells that have, these brush border enzymes. Okay, these are cells that carry the brush border enzymes. The brush border enzymes are basically these integral membrane enzymes that are sitting within the membranes, they're sitting within the plasma membrane of the simple columnar epithelial cells. And we see a lot of them within the jejunum. A lot of them. We have more of them within the jejunum. If you remember, the jejunum is where almost 90% of nutrient absorption will occur within the small intestine. So keep that in mind. So these brush border cells, these are your small intestinal cells. Okay? They contain the enzymes. They contain the digestive enzymes, the brush border enzymes. And these enzymes are going to further digest the disaccharides, are you following? Into monosaccharides. Remember, the monosaccharides are the simplest units of carbohydrates. Okay? So these organic compounds must be digested to their simplest forms before These simplest units can then be absorbed by your small intestinal cells. I hope you're following so far. Okay? All right. So, what are some of these brush border enzymes? Well, we have maltase. Remember, maltase is an enzyme that can break down maltose into two glucose molecules, and that can then be absorbed by your simple columnar epithelial cells. Okay? And then we have your sucrase. Okay? Sucrase can break down sucrose into one glucose and one fructose molecule. We have lactase. Lactase actually helps us with the breakdown of lactose. Okay, some of us do not have this enzyme, hence why we are lactose intolerant. All right, so lactase will help to break down lactose into glucose and galactose. Are you following? So remember, your small intestinal cells, especially the ones in the jejunum, they contain brush border enzymes, okay, on their plasma membranes. All right, this is why they tend to call them the brush border cells. Are you following? Okay, now the monosaccharides. I hope you remember from AP1. Here are the monosaccharides: glucose, fructose, galactose. They're the common ones. All right. They are now absorbed. Like I said, they are absorbed by your brush border cells, or you can say your small intestinal cells, especially of the jejunum. All right. And once that happens, the simple columnar cells, what they're going to do is they're going to pass on these absorbed monosaccharides. They're going to pass it on into what? Your blood vessels. Okay, so the goal is to take it back into the venous system, carry it to the hepatic portal-vein. The hepatic portal-vein will take it to the liver to process, and from the liver, it's going to go to the primary cells that need the glucose. Are you following? So this is usually the pathway. Okay, now keep in mind, sometimes all of the carbohydrates in your diet are not broken down because sometimes we lack certain enzymes. So carbohydrates that are not digested, they are called fiber. That's what we call fiber. And this is what will move down into your large intestine, okay? And that will get broken down by the bacteria in your large intestine. Are you following? And the large intestine can use that, those nutrients there to help us make some important vitamins, right? It's good for us, but the side effect is this can produce some intestinal gas known as, you know, the flatus. Okay, so it produces, this effect of flatulence. All right, so the gas produced by our intestine is actually coming from the bacteria within our large intestine. Okay, so keep that in mind. So once glucose, fructose, and galactose, remember these are the monosaccharides, once they have been absorbed by your intestinal cells, your simple columnar epithelial cells, from your small intestine, specifically the jejunum, once they have been absorbed, remember I said they will be now transported into the capillaries to be delivered to the liver. So the liver will receive them through the hepatic portal vein. Now, the liver will process these monosaccharides that have come from the digestive system. Keep that in mind. That is the importance of the hepatic portal vein. So once the liver gets these monosaccharides, it will, like I said, it will convert fructose and galactose into glucose. You're probably going, why is that, Miss Ogaga? Well, glucose is the cells prefer to use glucose compared to fructose and galactose. However, fructose and galactose, structurally, they're related. Keep that in mind. They're isomers of each other. Structurally, they're related. Their chemical formula is the same. But the way that they're arranged, their spatial arrangement and structure can be different. So that's why I say they're related, okay? Because the chemical formula is the same. So what can the liver do? Because the body prefers to use glucose. Well, the liver will convert fructose and galactose into glucose. Okay, so this is why I said the liver will be the place to process the monosaccharides before we release the glucose to the body cells. Keep that in mind. Okay, now once the liver has processed the monosaccharides, converted fructose, galactose into glucose, now we have what? A lot of glucose. So the liver then will now release the glucose into the bloodstream to be transported to your body cells. And your body cells will then use the glucose to produce ATP. Okay, so this is the general process. This is the metabolism of carbohydrates within the body. Are you following? Now, whenever we have excess glucose molecules, how will the body store it? Well, excess glucose molecules are stored in two primary locations. Keep that in mind. They are stored in the form of glycogen. Glycogen, remember that. All right? So excess glucose molecules are stored as glycogen, but where? There are two primary locations for this. They can be stored in your liver. It's definitely going to be stored in your liver. And there is another location for this. It can also be stored in your skeletal muscles. So these are the two primary locations for the storage of glycogen. Keep that in mind, okay? When you have excess glucose molecules, the body will store it in the form of glycogen. And the two major organs where you'll find glycogen, the first one will be the liver, and secondly, it will be your skeletal muscles. Don't forget that. I'm giving you the primary organs. You can find glycogen in other places, but in very, very small amounts. But the primary organs that will store glycogen, are you following me? It's going to be the liver, the skeletal muscles. So don't forget that. So now what I'm going to show you here are different processes involved in the metabolism of carbohydrates. So make sure you know these terms, make sure you understand what they mean. The first one is glycogenesis. Glycogenesis. So glycogenesis is actually the process that synthesizes glycogen. So I just talked about that. So glycogenesis is how we form glycogen from what? Excess glucose molecules. Keep that in mind. Okay? Excess glucose molecules. Are you following? So, and like I said, the glycogen can be stored in the liver and in the skeletal muscle cells. Okay? Another process, if we can synthesize glycogen, we can definitely break it down. So the breakdown of glycogen is what we call glycogenolysis. How you remember it is glycogen means, glycogen. Lysis is when you break something down, okay? So glycogenolysis is actually when the body breaks down glucose, okay? Glycogen, not glucose, glycogen is broken down into glucose. That's what I meant. All right, so glycogen is broken down into glucose molecules and released into the blood. Now, under which conditions would you want to do this? Well, let's say the glucose, the glucose supply of the body is depleted. We can usually see this. All right, some cells, such as your brain cells, they can only use glucose for energy, your brain cells. If you're low on glucose, it can affect your brain cells. They require a constant supply of glucose. So this is a perfect scenario where glycogen can be broken down into glucose for energy. Are you following? So where your glucose supply is depleted, where glucose levels drop in the blood, glycogen genolysis will begin, which is break down glycogen into glucose molecules and release it into the blood. Okay? All right, let's talk about another process here. Gluconeogenesis. Gluconeogenesis. So, what is gluconeogenesis? So, let's break it down. Gluco means glucose. Neogenesis is actually the creation or the generation of something. So this is where we synthesize glucose molecules from other organic compounds. Are you following? So, for example, lipids, which are your fats, right? And proteins. Yes, the body can actually do this. Oh, yeah. The liver can do this. Oh, yeah. It can take lipids and proteins, and from them, it can synthesize glucose molecules. So, this is the process called gluconeogenesis. Okay. This usually happens when our glycogen storage is what? Depleted. When we don't have enough glycogen, running out of glycogen from the liver. Are you following? This is when this can occur. The body will take your proteins, take your fats, and begin to now use them to make glucose molecules because it needs what? Energy. Your body cells are looking for energy. Okay? Now, there is a fourth process that I do want you to know, and this fourth process is actually related to cellular respiration. This fourth process is what we call glycolysis. Glycolysis. So, glycolysis is a process that involves the breakdown of glucose into ATP. ATP. So, this is a process carried out by all our cells. Okay, they use this to produce ATP. So this is the fourth process in the metabolism of carbohydrates, glycolysis, glycolysis. You are breaking down glucose molecules to produce what? ATP. Don't forget that. Okay, it is involved in the process of cellular respiration. And all cellular respiration is all about is how do the body cells take glucose from the diet, all right, on nutrients or anywhere else in the body, how they take the glucose, and then from that glucose, they can now make energy to carry out their essential what? Their essential functions, okay? Now, remember, glycolysis is an anaerobic process. It occurs within the cytoplasm of your body cells. It's an anaerobic process. Remember, something that is anaerobic does not need energy, not energy. It doesn't need oxygen. That's what I meant to say. Okay, so glycolysis, no oxygen required, occurs within the cytoplasm of our body cells. So when we look at glycolysis, what are the key players here? Okay, well, glucose molecules that like I said, will be converted eventually into ATP. But there are other processes involved, other steps. So what do we have here? We have glucose being a 6-carbon molecule. It is going to be broken down. Yes, you can say it can be oxidized, right? It's going to lose some hydrogen electrons in the process. But it is going to be broken down into two 3-carbon molecules of pyruvate. So pyruvate is a 3-carbon molecule. That's what pyruvate is. So from one glucose molecule, I'm going to get 2 molecules of pyruvate. Don't forget that. Okay? And there are additional steps involved in glycolysis. You don't have to know all the details. All right? So pay attention to the main things that I point out to you here. Okay, so because you can see right here that there are multiple steps involved. Look, this is the glycolysis pathway. There are multiple steps involved. It does involve putting energy into the system, meaning using up some ATP, and then also producing some ATP. Okay, but I want to draw your attention to this. At the end of the net gain of ATP from glycolysis will be 2 ATP molecules, the net gain. It will be 2 ATP molecules. Remember that, okay? And not only 2 ATP molecules will be produced, we will also see the production of the reduced form of that coenzyme that I talked about before, NADH. So remember, NAD plus is a coenzyme. It carries hydrogen electrons. And you're probably wondering, why is this important, Ms. Ogaga? Well, it is important because we're going to use these hydrogen electrons within the mitochondria eventually to generate even more ATP molecules. Okay, so keep that in mind. So not only are we producing a net gain, having a net gain of 2 ATP molecules, we're also carrying hydrogen electrons by reducing NAD plus. Remember, the reduced form is the NAD plus that has gained hydrogen electrons. So the reduced form will be the NADH. This is the reduced form of NAD plus. Don't forget that. Don't forget that. All right. Now, what happens to the pyruvate molecule that we have made through glycolysis? Okay. If oxygen is available in the cell, if oxygen is present, pyruvate molecules will move on to the next process in the mitochondria. Are you following? It will continue on to the next process in the mitochondria, and that next process is going to be the citric acid cycle. Keep that in mind. So I'll repeat that. If oxygen is present, pyruvate will then be moved into the mitochondria for the next process of cellular respiration. Are you following? That next process is the citric acid cycle. But if oxygen is not present, what happens to pyruvate? Well, pyruvate will be converted to lactic acid. And this is an anaerobic process because oxygen is not present. Your skeletal muscle cells usually can carry out this process. They can do this. Whenever the need for oxygen is very high, especially when you're doing a strenuous exercise, your skeletal muscle cells, they can take the pyruvate molecules that are being generated within the skeletal muscle cells, they can convert it to lactic acid. And of course, it is that lactic acid buildup in our skeletal muscles that will cause the soreness the following day or the fatigue within our skeletal muscle fibers. Okay, so keep that in mind. So here is an example of an anaerobic situation where pyruvate is being converted right here. Here's pyruvate. It is being converted into what? Lactic acid. Bacteria can do this. And like I said, our animal cells can do this. And also in this process, just the pyruvate to lactic acid, it will actually help to regenerate that coenzyme. Remember that coenzyme? The coenzyme was, reduced, remember, from glycolysis, we ended up with the reduced form of our coenzyme, NADH, okay? So this NADH can now be recycled and it can be oxidized to produce the NAD plus again. The NAD plus is required, for the glycolysis process to carry hydrogen electrons. So we have to regenerate it. Are you following? So this is usually what occurs here. Okay. So let's talk about the citric acid cycle. Here it is. So like I said, when oxygen is present, the citric acid cycle will be the next step in cellular respiration. And this is an aerobic process. It requires oxygen. And where exactly is it taking place? It is occurring within the mitochondria, but you have to go more specific. Remember, the mitochondria is a double membrane structure. Okay, it has a double plasma membrane, double layer. And you can see the outermost layer right here. All right, let me point it out to you. This is the outermost layer. And here is the innermost layer, the inner membrane and the outer membrane. Okay, and then we have, of course, the intermembrane space between the two. And then, of course, we have the inner space. The innermost space will be your mitochondrial matrix. Okay, so this inner space is what we call the matrix. And this is where the citric acid cycle will occur. So the citric acid cycle, it occurs within the matrix of the mitochondria. Do not forget this. Are you following? So what usually happens here in the matrix is the pyruvate from the cytoplasm, from the glycolysis process, will be shuttled in to the mitochondrial matrix. And that pyruvate will be converted into a 2-carbon molecule called acetyl-CoA. And then acetyl-CoA is going to undergo series of steps. It's going to undergo series of reactions to produce more ATP. Keep that in mind. Are you following? And in the same process, we are going to utilize our coenzymes. Remember NAD plus that I talked about before? Remember FAD plus that I've mentioned before? They are going to carry some hydrogen electrons from this process. And keep in mind, the reason they're doing this is because they're preparing for the last stage of ATP production, which will occur in another area of the mitochondria. Okay, so the citric acid cycle step will produce two ATP, a total of two ATP. Don't forget that, two ATP molecules. Okay, and not only just the ATP molecules being produced, we're also going to have what? The reduction of our coenzymes. So what I'm saying is they are going to carry electrons, and you can see that right here in this picture. Notice this is their reduced state. So we're going to generate some reduced coenzymes that are carrying our hydrogen electrons. And this is going to play a major role in the electron transport system, which is the last stage of cellular respiration. Okay? So what should you remember about the citric acid cycle? Remember, it occurs within the mitochondrial matrix. Don't forget that. Okay? It begins with the conversion of pyruvate into acetyl-CoA molecules. All right, and acetyl-CoA molecules will undergo multiple reactions, and one of those reactions that will occur will be the decarboxylation reaction. Remember this one, decarboxylation reaction. This is where CO2, carbon dioxide, is removed. It's removed in the process as these reactions of the citric acid cycles are taking place. Carbon dioxide will be eliminated. Keep that in mind. Okay, decarboxylation reaction. When you see that, you know, oh, this is taking place in the citric acid cycle. I get it. It's in the mitochondrial matrix. Okay, keep that in mind. Don't forget it. And remember, like I mentioned, your coenzymes, NAD plus and FAD plus, they are going to carry hydrogen electrons. They are going to be reduced into NADH and FADH2. Are you following? So all of them are going to carry hydrogen electrons. So at the end of the citric acid cycle, what should you see? Make sure you know this. All right? We're going to end up generating 8 NADH molecules, 2 FADH molecules. Remember the carbon dioxide elimination that I was talking about, the decarboxylation reactions that will occur? Well, they will produce 6 carbon dioxide molecules. And most important, 2 ATP molecules. I hope you remember that. So, based on what we've covered so far, let's quickly review. Glycolysis, which occurs in the cytoplasm of cells, produces what? A net gain of 2 ATP molecules. Citric acid cycle, which occurs within what? Within the matrix of the mitochondria, it will release what? 2 ATP molecules. Keep count of the ATP molecules. Are you following? And don't forget, we also, from both processes, we also have what? The reduction of NAD plus. These coenzymes will carry hydrogen electrons. Okay? All right. The third stage of cellular respiration is the electron transport system. The electron transport system. And this process involves something that we call oxidative phosphorylation. Oxidative phosphorylation. Oxidative phosphorylation is how we make ATP in the electron transport system. Keep that in mind. This is an aerobic process. This is an aerobic process. Okay? It requires oxygen. Where does it occur? Well, in the mitochondria, but in the cristae of the mitochondria. So Another term for that cristae is the inner membrane of the mitochondria. So let me go back and show that to you from our picture. You can see that right there, the inner membrane that is folded. Notice the folded inner membrane. The folded inner membrane is the cristae. The folded inner membrane of the mitochondria is what we call the cristae. All right, so that is where this process, this oxidative phosphorylation, is going to occur. And what does it begin with? Well, remember your coenzymes NAD plus and FAD plus? Well, remember they were reduced, right? NAD plus, we had some of them reduced in glycolysis. FAD plus and NAD plus, we had some of them also reduced in the citric acid cycle. When I say reduced, I mean they gained hydrogen electrons. They were carrying hydrogen electrons. So what we're going to see here is these coenzymes are going to deliver their hydrogen electrons to the electron transport system. Are you following? They're going to deliver it to the inner membrane, that cristae of the mitochondria. And this is how they're going to participate in the electron transport system. Okay? So remember this. When we look at the inner membrane of the mitochondria, the folded region, that folded inner membrane, the crista, what we're going to see is we have numerous proteins embedded within the membrane, and we call these proteins cytochromes. They're the cytochromes. So these cytochromes are the ones that are going to receive the hydrogen electrons from my coenzymes. Are you following? So they will receive these hydrogen electrons through some redox reactions. So what are we saying? NADH and FADH2, they are going to be oxidized. They are going to be oxidized. They will lose those hydrogen electrons. And you can see that right here in this picture. Here is FADH2. Here is NADH2. I mean NADH, not NADH2, but NADH. Here they are. And notice that they're delivering the hydrogen electrons to the cytochromes. These are the cytochromes. Here they are. These are the cytochromes. They're embedded within the inner mitochondrial membrane. So notice how they are delivering the hydrogen electrons to the cytochromes. And the cytochromes will then shuttle it across into the intermembrane space. Are you following? So what can we say about these coenzymes? Well, they're being oxidized. They're losing their electrons and giving it to the cytochromes to shuttle these hydrogen electrons into the intermembrane space. Are you following? Now, there is another major enzyme that is found within the inner mitochondrial membrane. It is the ATP synthase. The ATP synthase. It is this one, ATP synthase. The ATP synthase will be the main enzyme that will catalyze the production of ATP. It is the main enzyme. So what is going to happen here is, as these hydrogen electrons are shuttled into the intermembrane space, this concentration gradients, this proton gradients, remember hydrogen electrons are also called protons. Okay, so these hydrogen ion, the concentration gradient of this hydrogen ion is going to drive the production of ATP when the hydrogen ions begin to pass through the ATP synthase to come back onto the matrix side. So the flow of the hydrogen ions through the ATP synthase will now drive the production of ATP, and you can see that right here. So keep that in mind. Key things that are important. Your reduced coenzymes are passing on the hydrogen electrons to the cytochromes. Are you following? Once they do that, they're building up a proton gradient on the intermembranous space area. Okay? And eventually, the last enzyme, the main one that is going to catalyze this whole ATP production, we call it the ATP synthase. It is going to produce that ATP for us through what? The flow of the hydrogen ions, these hydrogen electrons. It's going to help it's going to use the flow of these hydrogen electrons flowing through it to now help to generate ATP. Okay? Now, something else you should understand is this. When the hydrogen ions flow back into the mitochondrial matrix, what are they going to be used for? Well, they are going to combine with oxygen. They're going to combine with oxygen. So oxygen becomes the final electron acceptor. Notice, oxygen is going to combine with these hydrogen electrons, these hydrogen ions, to produce water molecules. Are you following? So remember that. So oxygen within the mitochondrial matrix will be the final electron acceptor. Don't forget that. Okay? What we mean by that is oxygen will now bind to these hydrogen electrons to form water molecules. So the ETS, the electron transport system, all right, its job is to produce the maximum amount of ATP, and it is going to generate, are you guys ready for this? It is actually going to generate 34 ATP molecules, up to 34 ATP molecules. Okay, the ETS produces the largest amount of ATP molecules when you think about all the different stages of cellular respiration. And not only is it producing 34 ATP molecules, it also will produce oxygen molecules. Don't forget that, oxygen molecules. I already explained why. Because oxygen is the final electron acceptor in the ATS. Okay, we're producing oxygen molecules. Okay, do not forget the action of the ATP synthase. Okay, so products of the ETS are your water molecules, okay, and ATP molecules. All right, so remember, oxygen is your final electron acceptor. The electrons that are transported on this side are going to bind to oxygen. Once they bind to oxygen, we produce water. Are you following? So don't forget that. And we're going to produce 34 ATP molecules out of the ETS. Okay, and I'm telling you, this 34 ATP molecules are going to provide the maximum amount of ATP for your body cells to carry out their essential functions. All right. Now you're probably thinking, well, how did we calculate all of this to come up with the ATP molecules? Well, remember, we had two 8-ADH molecules from glycolysis. All right. That gives us 6 ATP. We had 8 coming out of NADH molecules from the citric acid cycle. That gives us 24 ATP. And then we had 2 FADH2 molecules coming out of the citric acid cycle that gives us 4 ATP molecules. So a total, all of your coenzymes that helped us in carrying hydrogen electrons, well, guess what? They helped us to generate 34 ATP molecules in the ETS. Are you following? So finally, I want to say this to you. For every glucose molecule, how many ATP will our body cells produce? It will produce up to 38 ATP molecules. And you already know majority of those ATP molecules are coming from the ETS. All right, remember, 2 from glycolysis, 2 from the citric acid cycle, and 34 from the ETS. Okay, for every one glucose molecule, our body cells can produce 38 ATP molecules. Are you following? And also take a look. We also produce CO2 from the citric acid cycle, water from the ETS. This is making sense. So this is the catabolic reaction of glucose. This is what breaking down glucose can produce. Okay? Now remember, 40% of the energy release is converted into ATP, which we've seen. 60% of that energy will be lost as heat within the body. Okay? So how do we regulate glucose catabolism? How do we do that? Well, we have two hormones that you need to know, and you're probably familiar with them already: insulin and glucagon. So insulin is produced by the pancreas, specifically the beta cells, all right, of the islets of Langerhans. You need to know this detail. You need to know it. And insulin is usually released by the pancreas when our blood sugar levels are low. When you have a low blood sugar level, your glucose level in your blood is very low. Is that true? No, that is not true. So let's rephrase that. I did that to catch your attention a little bit. So let's repeat that. When do we take insulin? People that have to take exogenous insulin. Well, when their sugar levels are what? High. Their sugar levels are high within the blood. Yeah, I tricked you a little bit. It's okay. All right. So insulin, let's go back. Insulin. Insulin is produced or released by the pancreas when our blood sugar level is very high. So the goal of insulin is to help reduce your blood sugar level. Okay? Insulin gives the cells the signal to say, hey, pick up glucose from the blood. Take up glucose from the blood so that the blood doesn't have an excessive amount of glucose in it. Are you following? So insulin will lower your blood glucose level to a normal state or a normal range. Are you following? And don't forget, insulin is released by your beta cells of the islets of Langerhans, which are found within the pancreas. Now, the opposite of insulin is glucagon. So glucagon has an antagonistic effect when you compare it to insulin. So when is glucagon released? So glucagon is released when your blood glucose levels drop, when they drop below the normal range. Are you following? Remember what I taught you. I said the brain is one of those organs. It has cells that are highly dependent on glucose. Are you following? This is why it is important to have a normal blood glucose level. Okay? So glucagon will play a role in keeping this homeostatic state in check. Okay? So glucagon, it is released when the blood glucose level is low, lower than normal. Okay? So glucagon is released by the alpha cells of the islets of Langerhans, which is also found in the pancreas. Are you following? And what will glucagon do? Glucagon will communicate with the liver. What's found in the liver? Well, glycogen is found in the liver. So glucagon will tell the liver in places like your skeletal muscles, hey, break down glycogen. Break down glycogen. Okay? So we have glycogen being stored in the liver and what? In the skeletal muscles. We know that already. So when we need glucose, when we need more glucose, we know to go to the storage site, which will be the liver and what else? The skeletal muscles. Are you following? So this is exactly where glucagon will go. It will go to your liver and skeletal muscles and tell it to, hey, break down glycogen. Release the glucose from glycogen because glycogen is actually a major polysaccharide made of what? Many units, chains of glucose. Are you following? So now the liver and the skeletal muscles, they can break down glycogen to release what? Glucose into the blood, and this will help to raise your blood sugar levels back to a normal range. Okay, so glycogenolysis, gluconeogenesis, the primary organ where they will occur is in the liver. Don't forget that it's in the liver, but it can also happen in your skeletal muscles because they have glycogen too.